So ever since humans have looked at the stars, they've always wondered, could we go to the moon, go to another planet, and look what's up in that sky? And so I want to walk you through a bit of some work we've done in our laboratory to help explore some of these places nearby on a planetary scale, and then also what's coming far, far in the future. So in particular, this plot of how the vision of getting to Mars and sustainably becoming an interplanetary species is a vision for humanity, for NASA, for many space agencies and many companies. And what's extraordinary, though, is this was really impossible until just the last few years, because if you look when Sputnik first launched in 1957, there was, of course, a bit of a space race between the USS and Russia, but only in the past few years have we broken through 70 years of relatively slow progress on launches, so that now there's something launching into space almost every single day. There is crew or a cargo or some experiment being launched into space. So now we have this really a second space race. It's not just the US and Russia, it's also all these other countries. So we have this extraordinary period where we can launch large missions and also begin to launch humans into space for some of these missions. And once you start getting people in space, of course, it raises questions of, what are the risks to the body? Of course, there are many, many risks that have been identified by our lab and many others. But we had a really unique opportunity to look at what happens to the body when you're in space for a full year. This was the case with the NASA twin stay, where Mark and Scott Kelly went on a year-long mission. Scott Kelly went in space for a year, and Mark Kelly stayed here on Earth. It's actually now Senator Kelly. So actually we have the beginning of maybe the senatorial genome project happening here. We could see the genetic differences between Republicans and Democrats, and maybe finally figure out what's happening for some of the government lockdowns. But I think we can actually at least understand what happens to the body in space. Politics will save for later. But we did find a few years ago we published a work of how many thousands of changes we observed across proteins, gene expression changes, metabolites. Many, many thousands of changes occurred in the body. But the good news is most of them reverted back to baseline. But this gave us the first map of every single molecular change that occurs in the human body and a way to figure out what happened for other missions. So we also then worked with some of the Inspiration 4 crew to do an all-civilian crew that went in space for an orbital mission and compared them versus longer missions and dozens of other astronauts to create this space omics and medical atlas. So just last year we published a large compendium of every molecule that we can see in human and mice that has ever changed in space, which has been extraordinary to see that most things, it's mostly good news, most things revert back to baseline, but it gave us this map of what's really changing in biology across this atlas. But I want to spend most of today to tell you about how we're moving from measuring biology to defending biology. Because if you look ahead, if you think about these other upcoming missions, Mars would be the hardest trip yet. You can see the amount, this is in log scale, the amount of radiation would be probably over 1,000 millisieverts, much more than what was seen for the twin study or the Inspiration 4 mission. This would mean a really tough challenge for radiation and survivability for the human cells. And so we've been working to take lessons from every creature we possibly can to protect cells. So in particular, a cute little creature like the tardigrade, this water bear, can survive in the vacuum of space and can survive huge doses of radiation that would easily kill a human cell. We've taken genes from that creature and put them into human cells, so now we can get about an 80% reduction in DNA damage. We take basically these tardigrade genes and put them into human cells. So you can see here some of the cells from lab that have this interchange between human cells and tardigrade genes, and it's functioning. And of course, you've seen in the news, of course, CRISPR methods that let you modify genes. But we've also been thinking in the lab about instead of just doing adding or subtracting human or tardigrade genes, you can also look at the fabric of life and think about epigenetic means to control how cells respond. So basically epigenetic tools to reprogram gene expression. In particular, there's been a wide range of methods published by many groups in the field to begin to build this tool set of imagine if you could reach inside of a cell and turn on a light switch, turn on a gene or turn off a gene, have this exquisite control for each gene that you would want to activate or repress. And so working in the lab now, we actually have these specific target genes that you can turn on or turn off. Here's 6 examples, 3 of each, that we've had working now in culture. So this ability to reach into a cell and control genes at very specific sites, which I thought was a fantasy when I was a graduate student, is now something that's actively happening in my laboratory, which is extraordinary. So if we can do this, you can start to think ahead a little bit and think, okay, what about future missions? If there really is a space station around the moon, what's here is called Mars Base Camp, could we think of what would you need to survive if you're going to bring all the bearing and tools of molecular biology to survive? So we could have a couple ideas. I think one of them, for example, would be, if you think about in your genome, there's actually a gene that makes vitamin C, but it's been degraded. It's called a pseudogene. But in your body, it's still there. And so instead of getting vitamin C, for example, from like margaritas, which is a totally legitimate way to get vitamin C, that's fine, could you have it just be made in your own cells? We know that, for example, across evolution, multiple mammals have lost it, but some have kept it. So dogs and cats don't get scurvy. They can still make their own vitamin C. And the gene for this is still inside of our genome. It just has to be a bit tweaked. And while we're at it, we could think of, okay, for long missions, you want to get vitamin C, it'd be nice to make it by yourself. But what about other things you need? You need 20 amino acids, but 9 of them we have to get from our diet. We can't make them. Well, why is that? Wouldn't it be nice to make all of your own amino acids? I feel very sad about my amino acids if I can't make them all. So I'd like to make them all. But now we know we can complete the genetic circuits by using genes from other organisms and complete the circuit within mammalian cells to make it so you could become this basically autonomous human cell that makes all your own amino acids with this completed circuit. And while we're at it, we could think of other targets. So, okay, if you're thinking these long missions, what about, for example, repairing disease genes, making it so you could have control of hemoglobin or of a lower all risk of any disease? And this also seemed hypothetical 5 years ago, but now there's been CRISPR therapies to cure beta thalassemia and also sickle cell disease that went from a clinical trial to about 3 years later an FDA-approved therapy very quickly. So this idea of CRISPRing to prepare disease genes went from science fiction to science fast really quickly, and has all just more recently been deployed actually to help with the CPS1 deficiency in a newborn even. So it's a really extraordinary time where we now have effectively cellular liberty. You're no longer beholden to the genome you got at birth. You can actually go in and reprogram your cells as needed to repair a disease or maybe to prepare for long missions. And that's true for human cells, but also for microbial cells. When we look ahead, we want Mars to have this total autonomy. We want Mars to become self-reliant. So the microbial species, fungal, plants, animal cells all have the capacity to make their own drugs, make their own survival kit effectively in their own cells. And so we're continuing to look at, this as a model, but also look at other places all over the world where we see extremophiles that have survived and adapted to learn from them. Every creature, every genome carries a genetic payload that has lessons, evolutionary lessons that have been hammered out through billions of years that we can learn from in any and all of these environments. And so someday, could we have plants that are on Mars that look like this? Well, potentially. And could we, with new launch capacity, imagine missions that go, for example, to Mars and Venus and back again, all within about 18 months? And indeed we can, because this is in the second space age. It means we can go farther, we can go faster, and we can go with bigger rockets and with more payloads than ever before. Which then gets us to the stage where we could have basically planetary liberty. Instead of just being on one planet, which Earth is great, but we could begin to go back and forth to Mars. And could you have genes that you activate on your way to Mars and reactivate some other genes on your way back to Earth? Could you have another planet that you could go to and have planetary liberty? And I think eventually we can. And then if you're thinking you do this solar system, what about elsewhere? Where else could we go? How many exoplanets are there? Planets around other solar systems. There are now, it used to be 0, but now there are thousands of exoplanets. Several hundred of them are likely in the habitable zone where there might be liquid water that are relatively close to us. You can see here some examples that are a few light years away, not that far. And we've actually mapped out and seen for every exoplanet that's been found that potentially has habitable, which you can see here is either blue or purple, in our galaxy from the top or the side. These are all the planets that are relatively close to us in a galactic sense that we could go to, maybe even survive relatively soon. So they could imagine in the future making a generation ship that has multiple generations living and dying on the same spacecraft on the way to a star, and you hope that when you get there it works out well, otherwise you've got to go to a different planet. But this is complete science fiction 100 years ago, but is now being actively planned for by several agencies and research groups, and is something that's very possible to do today. But I want to close with an important answer to a question I almost always get, which is, well, just why? Why spend all the time, energy, money doing this kind of work when there are diseases, there's poverty, there's other problems in society, there's many, many problems you could be addressing. Why spend time doing this? And I want to really address that head on. There's at least 3 good reasons why. And the first one is that you can make progress in both science and society at the same time. We can, for example, land people on the moon and pass civil rights legislation. We know we can do both things at the same time. The second reason is an intangible asset of hope. Is that this example of our daughter when she's out running into the ocean, and then I talk to her about looking in the future, and we go to Venice Beach, and you go down to like a skateboard park. But we had an actual discussion about what a skateboard park would look like with 1/6 gravity on the moon or 3/8 gravity on Mars, and she could imagine herself as a lunar architect, which is it's an actual job. So it's extraordinary to think about this world that's not hypothetical, but is a future world that the future is brighter than the past, has more potential than the past, and that's a world I think she lives in. I want to make sure that she still lives in. And the third reason, perhaps the most important, is a sense of duty. Actually, life, as far as we know in the universe, is extremely rare, it's very precious. In fact, for example, in North America, we've lost about 1/4 of all birds due to habitat destruction, already in the past really few years. And we know that microbes are being lost before we can even sequence them. They're being disappearing before we can catalog and characterize them. And this is not the first time. If you take the last 500 million years of our Earth's history and then look at the temperature, we've had a world without polar ice caps and a world with polar ice caps. And it's fluctuated many times over the past 500 million years. And I think about this chart almost every day when I wake up. It's the thing about this history. Of course, we're trying to avoid what has happened every time a rapid temperature change occurs. We have mass extinctions, at least 5 so far. We would not like to have another one, and we're trying to avoid that right now today. But even if you take a step back, about 700 million years ago, we had a snowball Earth. And actually, if you take this 500 million years and you kind of zoom out and put it in the history of our planet and likely future, we're right about here, which is this very nice but temporary temperature canyon. At one point, the Earth cooled, and we've got this nice liquid water, but eventually the Earth will get hotter, it'll be engulfed by the sun. When I was a kid, I always thought that we had about 4.5 billion years until the sun was engulfed by the Earth, and I was very calm by that. But I was doing updated research, and about 1 billion years, actually, the Earth's oceans will probably boil as the sun's luminescence increases. And as I got really sad about this, and I was moping around my house, and my wife asked me, "What's wrong?" And I said, "I just thought we had more time." And she laughed, as you just did, but I mean it. Like, if you thought you could be 100 years old and all of life and all the beauty and complexity of everything on Earth would have billions of years and you get cut it by, 4/5, it was devastating. And I thought we could, we don't have as much time, actually. So I think, like, what's really at risk here is it's really, of course, consumers, all kinds of life are at risk, but I argue to you that there's a fourth kind of life, the kind of life that is really aware of the frailty of ecosystems and the value of life, is really the fourth kind of life, which is us, that can really serve as guardians and protectors of life. Quite literally, we are the guardians of the galaxy, I'm telling you, because only humans are aware of extinction. Only we can manifest this duty or even be aware of it all, which I think gives us a duty towards all life, past, present, and future. And even the past, which seemed hypothetical until recently, this idea of a duty towards past life has come up more recently, of the idea of, should we bring back the woolly mammoth, for example, which is being worked on, and work also with the board at Colossal Biosciences and their SAB to help them. And just recently, even the dire wolf came back, which was an extinct species, but it's now back, and some of its alleles recreated again. So this completely hypothetical world from 5 years ago is now science fact in our hands, which is an awesome and terrifying responsibility, but an extraordinary opportunity to actually think how we could engineer life and have it to protect life and preserve life and even bring it back sometimes. But then looking ahead, just to close, a closing thought, you could imagine, okay, fine, Chris, well, we're going to go to the next star system, and the next star system, just ad infinitum. What are you going to keep doing? Just keep going to new stars endlessly, just trying to preserve all life across the universe. What do you do when it's the big crunch or the big rip of the universe? What's going to happen? Just keep going and going. And I think here we have the final question, really, is, do we allow the universe to have its heat death or big rip, or have the universe end? Or do we try and alter the universe in some way so that we prevent it and actually make it so we could not have the universe die, or hope that life might arise again? And I think between these two options, we don't know if life will ever occur again. Is this the only universe where ever life did occur, will occur, might ever occur? And maybe the answer is yes. And even if the answer is maybe, I think we'd have to fight for it. I think we'd have to presume that we have to maybe try and restructure the universe itself at some point to be able to survive. And to do that, we'll need all the tools and mechanisms of molecular biology, engineering, tools of computational methods, and begin to create. And of course, we have to do this together, slowly, carefully, responsibly. But I think we have no other choice. Thank you.